fourth learning objective is considering what happens if we look at more than one character at a time. So we talked about yellow and green seeds, but what happens if we add in a second trait, smooth seeds and wrinkled seeds? So if we have a plant that is heterozygous for both of these traits, crossed with a plant that is green and wrinkled seeded, or has both of the recessive characteristics, what proportion of the offspring would we expect to have green wrinkled seeds? Okay, so before we can actually answer the question, let's talk a little bit about what happens when we have two different characters. So remember, with the purple and white flowered peas, Mendel is able to demonstrate the law of segregation, that when the parents produce gametes, if they have different forms of a gene or different alleles or different traits for a character, when they pass them on, those two different versions are going to separate, and then at fertilization, you're going to have one copy of the trait from each parent. So the F1 generation in Mendel's experiment were monohybrids that were big P, little p. They were all purple, but they all had the white flower gene in them. So we call that a monohybrid cross, one hybrid. But then Mendel wanted to take it a little bit further and said, okay, well, we've seen that the traits split up during gamete formation, and they come back together when a new offspring is formed. But what happens if we've got more than one character that we're looking at? So he had true breeding parents for two different characters, for seed color and for seed texture, whether it was round or wrinkled, and these were dihybrid crosses. So they have two different characters that were being looked at. And the question is that he was asking do these characters get transmitted or given to the progeny, the offspring, together as a package, or is it possible to separate them? Are they sent out independently? So in his experiment, which now we would represent as being controlled by two different genes, the yellow and green gene being represented by Y, the round wrinkle gene represented as the letter R. Okay, so the true breeding parents, which were yellow round and green wrinkled, were crossed together, and what do you get? And the F1 generation, all of them are going to be have the dominant trait being expressed, so they're all going to be yellow and round. Okay, so here is where things got interesting, is in that F2 generation. There were two possible outcomes that he would have expected. One is if there's dependent assortment. If those characters are always going to be transmitted together, then he would expect that the roundness and the yellowness would always go together, and the wrinkledness and the greenness would always go together. But that's not what he observed. What he actually observed was that in the progeny, he got all different combinations of roundness and wrinkledness and yellowness and greenness. Well, I say all different combinations, but he saw this ratio of 9 of the double dominant to 3 of the single dominant to 3 of the other dominant character to 1 of both recessive characteristics, or 9 to 3 to 3 to 1. That's a very sort of famous ratio, and hopefully you understand what it means. What it means is that there is independent assortment. You can separate the yellowness from the wrinkledness and the greenness from the roundness. So this just shows again what we now understand to be the genetic background behind this. When we cross this Y gene, when we look at the Y gene and the R gene, are they being allocated to progeny separately or together. And what one hypothesis suggests was that they happened together. A separate hypothesis was that they would be independent of each other. The yellowness was independent of the wrinkledness. And that's what he observed. So Mendel called this the law of independent assortment, which states that a pair of alleles, or the different traits for different characters, are going to segregate from each other independently during gamete formation. Now, we know that this happened for him because those genes were on different chromosomes. So they were not on the same chromosomes, or this phenomenon called linkage would have occurred. And we'll talk more about that in the next chapter. Genes can be physically located on the same chromosome, and they tend to be inherited together more often than not. Okay, so if we get back to this question of what happens if we cross a heterozygous green wrinkled plant, a heterozygous yellow smooth plant with a green wrinkled seeded plant, what proportion of the offspring would we expect to have green wrinkled seeds? And we can do another Punnett square, but we have to do a little bit differently because now we have different combinations of the characters and the traits, the genes, the alleles. So a green wrinkled seeded plant is only going to be able to give out these type of gametes, little y, little s. But a heterozygous plant can give out any of these 4 different combinations of gametes because they have all 4 of these genes present, 4 alleles of these 2 genes present. So when we draw out our Punnett square, this is what it looks like. We get big Y, big S, little s, all the way down here, because remember this parent is always giving the same thing. Big Y, little s, little s, little s. So these are going to be yellow and wrinkled. Little y, big S, little s. These are going to be green and smooth. And then little y, little s. Or green and wrinkled. And you can see that because all 4 of these are the same, the pattern is going to continue down these columns. Hopefully you can see that. What that means is that we're going to get 1/4 yellow smooth, 1/4 yellow wrinkled, 1/4 green smooth, and 1/4 green wrinkled. So what proportion of the offspring would you expect to have green wrinkled seeds? The answer we're looking for is 1/4 in this particular case. But if I were to mix it up and ask you a slightly different question in the context of an exam, hopefully you'll be able to draw out the Punnett square and determine what might be happening in a different situation. All right, this slide is just showing how this is not actually a P karyotype, this is a human karyotype. But here's what I was saying, that the genes that he was looking at for the roundness and wrinkledness and the yellow and the greenness were on different chromosomes. So they do assort independently from each other. If they had been on the same chromosome, so if that yellow-green gene was here on chromosome 1, same as the round wrinkled gene, things would have looked a little bit different. And that was something that was figured out in the early 20th century by a fellow named Thomas Hunt Morgan. But we'll talk more about Morgan in the next chapter. So moving on to a little bit past Mendel, here are our learning objectives for part 2. Okay, so now we're going to move a little bit beyond what Mendel found and sort of see how variations on what Mendel observed have meant to the study of genetics. So Mendel had a case where it was all or none, purple or white, and nothing in between. Now That was kind of how he wanted it. But as it turns out, in some cases, we can have two traits. We can have three traits, but it's controlled by a single gene. And we can have three different appearances, three different phenotypes. And the phenotype is always going to tell you what the genotype is. So this happens in a number of cases in plants, where if you cross red flowers and white flowers, you get pink flowers. Now, this sounds kind of like blending, but it's not, and I'll explain why. So, this situation is called incomplete dominance, when the heterozygote has an intermediate phenotype between the two homozygous phenotypes. So the appearance of the heterozygote is different from either of the homozygous parents, and the homozygotes look different from each other. Now, is this blending? No, it's not, because there's only one trait that is between the two homozygous traits. So if we cross the heterozygotes with the homozygotes, we don't yield more different gradations in the color. For example, here's a case from another textbook with purple eggplants and white eggplants. They can be crossed, and they're going to produce these sort of pinkish violet eggplants in the F1 generation. But then what happens when we do that F1 cross? Well, we end up getting some purple eggplants, half violet eggplants, and 1/4 white eggplants. So we don't get all violet, which we might expect under a hypothesis of blending. We get this 1 to 2 to 1 that we saw in the purple flower plants, only with an intermediate expression of the heterozygous phenotype. Okay, so what would happen if you crossed violet and white eggplants? So here's a Punnett square that shows that. And what we end up getting is half violet eggplants and half white eggplants. And you can see the genetic basis for that. If we had a blending going on, it wasn't a particulate case, then you would expect that crossing violet eggplants and white eggplants would give you a lighter shade of violet, something between violet and white. But that's not what we observe. So still, we have a particulate mode of transmission of hereditary information. So it's not blending. So here's another example with the same basic idea. Often snapdragons, which are a very common annual plant that people like to grow around here, have this going on, where if we cross red-flowered snapdragons and white-flowered snapdragons, we get all pink-flowered snapdragons. So if we go back to the original question, if we cross a red plant with a pink plant, knowing the genetic basis, what proportion of the offspring would we expect to have pink flowers? Okay, so again, the red parent can only contribute a big R version of that gene, the big R allele. The pink flower can contribute either the big R allele or the little r allele. So the progeny will either be red, big R, or pink, big R, little r. And what proportion of the offspring would have pink flowers? Well, 2/4 or half are going to be red, and 2/4 or half are going to be pink. So 1/2 red, 1/2 pink, and no white flowers.